Okay, so now we're going to look at this homeostasis in mammal. Okay? We use a mammal as our model to study about this homeostasis. And what we're going to look at here is we want to discuss the importance of the homeostasis. Why we need the homeostasis, okay? And we need to explain the principle of homeostasis in terms of few terms that we have to use here: internal and external stimuli, receptor. Very important for receptor to actually receive this stimulus. Okay, when detect the stimulus, then they have to send the input to the central control. Okay, central control, and this central control are going to send a message, okay, to the effector. So normally effector will be our muscle and glands, but in terms of communication between the receptor, central control, and also the effector, we need the coordination system, which we're going to learn a little bit more detail when we go to the final topic. Okay, so these are the first thing we're going to learn, and then we need to define the term negative feedback. So what's meant by negative feedback, and explain how it involves in the homeostatic mechanism. Okay, so we have two. We have negative feedback together with the positive feedbacks. Okay, but for homeostasis, generally will be negative feedbacks. Okay, so these topics, as I said, that you have learned it before. So here we just touch up a bit or add in a bit more detail in terms of the process. Okay, so homeostasis is a state of steady internal conditions maintained by the living thing. So highlight the words internal conditions. So means the external condition tends to change our internal condition. For example, very simple, when you have lunch, okay, high in the carbohydrates. So it means that your blood glucose increase. So when your blood glucose concentration increase, we cannot let it continue to increase. What we want to do, we want to bring it back. So this is what we call as the homeostasis. So no matter what kind of homeostatic control, we do need to have a control system. We need to have the receptor to detect the increase in the blood glucose level. We need to have the control or central control to actually give out the information, say, yes, we need to control. And then someone have to do the correction, so which is the effector. Okay, so three of them work together to achieve a steady or constant internal condition. That is meant by homeostasis. Okay, so it depends on the body ability to detect the stimulus and oppose the changes caused by the stimulus. The stimulus can be external stimulus, for example, okay, external stimulus, for example, you detect the heat. You touch your hand, you put your hand, you touch on a hot surface. Pain, something that causes you to have pain. So this is what I call external. Internal stimulus, for example, blood pH. Can you see that? Water potential of your blood. So these are the stimulus. So we have to respond to this stimulus and do the appropriate correction. So this is homeostasis. We want to maintain a steady internal or constant internal condition within a particular set range, okay, or set point. So this dynamic is a dynamic state. It won't be constant all the way, but it will fluctuate around the constant range. Okay, so in our syllabus, we're going to focus on these three: blood glucose concentration, water potential of the blood, and also the core body temperature. We're going to focus in these three in terms of mechanism, but don't forget, we also look at this metabolic base, particularly you have the metabolic waste, okay, particularly carbon dioxide and urea, how we regulate our carbon dioxide, how fast we need to exhale out the carbon dioxide. Okay, blood pH, as I say that we have to make sure is in the narrow pH, because if the pH too high or too low, it's going to affect our proteins, okay, the function of protein, the concentration of the blood of the respiratory gases, for example, oxygen and carbon dioxide. Okay, so we look at the effect of the physiological factor here. If the concentration of glucose in the tissue fluid is low, we know that if the concentration of glucose in the tissue fluid is low, then the rate of respiration is low or even stop. Then the cell will lack of the ATP. If the concentration of glucose in the tissue fluid is high, what will happen? it will cause the water molecule to be drawn out of the cell by osmosis, because outside of the cell in the tissue fluid, concentration of the glucose is high, means that you're going to lower down the water potential. So when the water potential is low, dense water molecule will be drawn out by osmosis into the tissue fluids. So it means that the cells will lack of water molecules. Then you can see that shrinkage of the cell take place. So how about water potential of the tissue fluids? So when the water potential of tissue fluid is low, again, low water potential, it will cause the water molecule to be drawn out of the cells by osmosis. So it will stop or slow down the metabolic reaction, because hydrolysis process require water. Okay, but if the water potential of the tissue fluid is too high, what will happen? Right, high water potential in tissue fluid, low water potential inside the cells. Therefore, water molecule will be drawn into the cell by osmosis down the water potential gradients. The cell may experience the bursting, okay, or lysis. How about core body temperature? If the body temperature is too low, we know that metabolic rate is lower because of the enzymatic reactions. Kind of depends on the temperature. Okay, the optimum temperature, for example, most of the enzymes work around 37 to 40 degrees Celsius, but now your core body temperature 34, 35. So metabolic rate will be lower. Okay. How about if, let's say, core body temperature is too high, it may cause the protein and enzyme denaturations, or the enzyme proteins cannot function, because it loses the precise three-dimensional shapes. Okay. Blood pressure. So blood pressure, if too low, as I said, that you try to imagine the blood pressure is too low, the blood may not be able to reach your brains. It may cause the dizziness. It may cause some part of your body may not have adequate blood supplies. Okay, but too high value, then what will happen with blood pressure? It may rupture our capillary. Let's say it rupture the capillary at the brain. It may cause the stroke. Okay, or the rupture of the capillary may cause the heart disease and also the heart attack as well. Okay, because your blood pressure is too high. Okay, so increase the risk of the potentially life-threatening health. How about the blood pH? So if the pH is too low, it will cause the acidosis. High pH will cause alkalosis. Both of them will cause the denaturations of the protein and enzyme. So again, the cells will lose the enzymatic reactions, or metabolic reactions. Okay, in this case, definitely it will bring us the disastrous effects. Okay, so guys, for us, highlight these three. We're going to focus in these three when we discuss the homeostatic control. Okay, blood pressure and blood pH, we won't talk about it. Okay, in these topics. Okay, so all homeostatic control mechanisms have at least these three interdependent components. Okay, so we have the receptor, control center, and also the effector. Okay, so what are their roles? For receptor, very important. They have to detect, they have to sense and detect, okay, and monitor to the change in the environment, either external or internal environment. So they must be able to detect. When they don't only detect, like, I mean, let's say, for example, in the COVID, I mean, you detect only, we didn't send the information, useless. Okay, so we have to detect, and they have to send what? Send an input to a control center. So two roles, receptor. They have to detect, and they have to send the input, or to send what we call this, the detail, okay, to the control center. One example of receptor is a thermoreceptor, the one that detects the change in the temperature. So what's the function of control center? So control center actually set the maintenance range. So they have their own range. Okay, from 1 to 2. Anything more than 2 is not acceptable. Or anything less than 1, not acceptable. So what we do, if the corrections, okay, or corrective measure must be done, then the control center will respond to the signal by determining an appropriate response and sending the signal to an effector to do the corrections. And this signal we call it as the output. Are you clear? You will send the signal, so the signal is known as the output. So for example, our hypothalamus is a highly integrated control center. Hypothalamus involved in many homeostatic process here, okay, as a control center. And the last one, we look at the effector. So effector is the one that receives the output, okay, from the control center and produce the response or responses and to change the physiological factor. Okay, so they have this ability, these have the ability to actually change the physiological factor. For example, let's say body temperature too high, then what we do, our muscle or our glands, sweat glands, release the sweat. So when the sweat glands release the sweats already, then what will happen? Cooling effect, bring down our body temperature. Okay, so for example, our muscle and also our sweat glands. Okay, so when a signal is received and acted on, so negative feedback is provided to the receptor that stop the need for the further signaling. So always remember, you can see that in the negative feedback mechanism involve the receptor, control center, and also the effector. So receptor, we're going to send the input to the control center. Control center, we're going to send an output to the effector. So effector will bring down or increase, either increase or decrease the parameter. So the increase or decrease of parameter will be detected by the receptor again. So it forms a loop. Can you see that? It forms a loop here. So basically, you need to inform, right? Okay, whether corrections have been made or not. If you didn't inform, what will happen? Then it will continue to cause the correction. The signal always sent to the effector. We try to imagine that our body temperature is high. We bring it down. When it's normal already, and there's no signal or no feedback mechanism to tell the receptor, because the receptor is still thinking that, oh, our body temperature is still high, it will make this deviate more from our normal range. So that's why feedback mechanism is very, very important. Okay, but you have to know that to send the input or output, we need a coordination system, which is either our nervous system or endocrine system. Nervous system, nerve impulses or action potential. Endocrine system, hormones. Okay. So what is the stimuli? Okay, stimulus or stimuli, they are detectable change in the internal external environment. So the ability of the organism to detect or respond to external stimuli is known as the sensitivity. Okay, sensitivity. So as I said, that those information, the transfer of the information require two systems here, either nervous system or endocrine system. So a general differences between the nervous system and endocrine system is summarized in this table 18.3. So in terms of the signal or the messenger, so for nervous system is the electrical messenger, so in the form of action potential. Endocrine system will be in chemical messenger. We call them as the hormone. So the organ that involved here for nervous system definitely involve the brains and the spinal cord, which is part of the nervous system. Endocrine system, it will be glands that involve in the endocrine system, which secretes the hormones. In terms of the cell types, nervous system, neurons, it will be involved. Endocrine system, then epithelial cells. The specialized epithelial cells form the glands that can produce the hormone and secretes the hormone. So transmission of the messenger, so through the neurons, the transmission through the neurons. Hormones will be transmitted through the blood vessel. So this one, we're going to learn it in detail tomorrow, right? When we look at how hormone functions, okay? So how about mechanism actions? So action potential make use of the neurotransmitter at the synaptic cleft and the sodium-potassium channel and enter the target cell. So this one, again, no need to worry at this moment because we're going to learn it in detail next topics 19, okay? And hormones maybe enter into the target cells by diffusing through the plasma membranes or by binding to the cell receptor, okay, at the surface. It depends which kind of the hormones. It can be either lipid-soluble hormones or water-soluble hormones, okay? So again, this one we're going to learn tomorrow when we look at how hormones actually give us the effect. So the kind of response, you can see a localized response. Localized response basically means target a particular system. For example, sweat glands, your skin. Okay, so localized response. Okay, but if you look at the hormones, hormones can be widespread. It can be target many organs like adrenaline can target, okay, widespread, okay. So the rate of response is quite quickly respond to the stimuli, means that it will within a few milliseconds. Are you clear? Few milliseconds. Now, even though for the hormones respond slowly, it doesn't mean that very slow until few years. It can be until few years, but sometimes even needs the quick one within few seconds, you can see the effects already. For example, when you look at a crisis, when you flip the question paper, something I didn't learn before. When you're nervous, crisis, then your heart pump fast, adrenaline rush in this case, right? So adrenaline rush, then you see that the effect will take place. It's not like immediately, it may be a bit like 1 to 2 seconds, okay? But some hormones work slowly. For example, growth hormone, okay? The growth hormone release, you won't see that one day increase by, a 10 centimeter, it won't be, okay? The increase will be quite slow, maybe few days, okay? You will see the effect, okay? For few years, you see the effect. So it depends on the situation, but relatively will be slower compared to the nervous system. Okay. Then the last part, we're going to look at this feedback loops. Feedback loops. So we want to continuously to monitor the changes, then so that we can have the continuous adjustment. So this one we need to depends on the negative feedback loops. Okay, negative feedback loops. So basically, just now I say that if I done the correction, you have to report to me. If no, then I do not know, okay, whether the corrections have been done or not. So receptor have two functions here. So receptor have to detect any changes and then send the input to the control center. Okay, so it minimizes the differences between the actual value if we use a negative feedback loop. So it means that the factor going to fluctuate around a particular ideal value, or we know it as a set point. So it's not really a value, but it's a range, and basically it's a range. So example, a homeostatic mechanism maintained by negative feedback loop will be blood glucose concentration, water potential, and core body temperature. One thing I'm going to add in here is the blood pressure, which is not in our syllabus. So as I say that our learning model will be blood glucose, water potential, and core body temperature, but it doesn't mean that only these three. Okay, it doesn't mean that only these three. We have more than that. So one of the examples is blood glucose. Okay, blood pressure that we're going to discuss now. So look at this. What it means that negative feedback mechanism. Okay, negative feedback mechanism. So let's say blood pressure increase above the normal range. So blood pressure increase may due to two factors. One, cardiac output. Okay, cardiac output basically means that your heart pump more blood into the blood vessel. When cardiac or your heart pump more blood into the blood vessel, then it increase the hydrostatic pressure. That's one. Or second, your heartbeat too fast. Okay, your heartbeat is too high. Okay? So what we're going to do here is you can see that we have the baroreceptor. Baroreceptor is the receptor that detect the change in the blood pressure. When there is a change in the blood pressure, what we're going to do here is this baroreceptor going to send to three control center. One is called cardiac acceleratory center. Another one is a cardiac inhibitory center, and also vasomotor center. So three of them. So this will be our input. So the input will be sent. So this input, either you can see that this sign means negative, arrow means that stimulate. So what we're going to do here is we inhibit the cardiac acceleratory center, but increase the cardio inhibitory center. So inhibit what? Inhibit, so decrease the cardiac output. So your heart will pump slower and then decrease the volume of the blood pump into the blood vessel. Okay, then how about vasomotor center? When we inhibit the vasomotor center, it will cause the vasodilation. So it means that increase the diameter. When you increase the diameter or arterial or the artery, then we will be able to reduce the blood pressure. So when the blood pressure decrease already, then you must be detected by baroreceptor again. If there is a decrease in the blood pressure, but it's not detected by baroreceptor, a continuous input will be sent to the control center. Then it will cause a problem. Vasodilation too much, then cardiac output too low, it may cause the blood pressure too low. Can you see that? So it won't oscillate around the what we call the range. Are you clear? So blood pressure reduced already, we need to send the what we call the must be able to detect by the baroreceptor. So similar, if let's say blood pressure too low, then the opposite way activated. So it means that you can see that vasoconstrictions occur, increase the cardiac output, bring the blood pressure back to the normal. Okay, so you can try to answer the questions here. Okay, so I go slow, so it's recorded the answer. Okay, so involve the sympathetics and parasympathetic. There is a little bit of problems here because in our syllabus, there's no sympathetic and parasympathetic nervous system will be discussed. Okay, now to compare or to contradict with the negative feedback mechanism, we do have what we call the positive feedback loop. Okay, positive feedback loop. Now, positive feedback loop is basically means that a deviations in the parameter from the ideal set point leads to a further increase in the deviation. So usually it will amplify the conditions that do not happen very often. Okay, so the conditions or the situation will continue until it is interrupted by some mechanism. For example, contractions in the childbirth, lactations, and also the blood clotting. So we're going to look at this contraction in the childbirth. Okay, so why we need to use a positive feedback mechanism? If you look at the process here during the labor or the childbirth, the delivery, so we do know that the uterus start to contract. When the uterus start to contract, the fetus here, okay, the head of the fetus now press against at the cervix opening. So cervix is a muscle. So cervix do have the sensor receptor, send the signal or send the input to the control center and tell the hypothalamus there, oh, delivery, or the childbirth need to start. So this is a positive feedback mechanism because one of the hormone will be sent to the effector, means the wall of the uterus is the oxytocin. So this oxytocin will cause the muscle to contract even more. Can you see that? So uterus start to contract, make this head of the fetus press against the cervix. The cervix will feel the stress. So this stretching will send, detected, send the input to the control center. Control center know that, oh, now is the delivery. So send more hormones to the effector, which is the wall of the uterus. What will happen here? The wall uterus now contract even more. So when the wall of this uterus contract even more, what will happen? The fetus now will exert more pressure to the receptor here. So this pressure will be detected by the receptor, and then control center continue to release more oxytocin. Can you see that? It will increase the deviation now to make the cervix dilate until the entire process finish. And luckily, it's not the negative feedback. If a negative feedback, what will happen? The cervix want to open, and then the brain asks to close. The cervix want to open, you ask it to close. Then forever, childbirth won't take place. Are you clear? So in this case, you can see that it's a feedback loop, but it's a positive feedback loop because the cervix dilations, if you look at this, the parameter, if this is the time, you can see that it continue to increase for the cervix opening, okay, until the entire process finish, okay, then it will go back to normal again. So this is a positive feedback mechanism, okay. So you can try the questions. So this is the answer here. And then another one you have, we didn't discuss here is the lactations. Lactation is when the baby, okay, sucks on this the nipple. So it will stimulate the nerve response into the spinal cord, and the hypothalamus then it will produce more prolactins and more milk for the baby. So the more babies suck the nipple to get the milk, the more milk will be produced by the mom. Okay, so this is the positive feedback mechanism. Okay, so with this, I have done for the principle of the homeostasis. And tomorrow, we're going to continue with a specific example, which is the blood glucose regulations. Okay, so with this, I've done for today. I stop the recording.